yeah, thank you so much for the invitation to present as part of this the CST Red Hot series. So I'll be presenting to you, as Rahul mentioned, on AI and research, specifically in transplant research, the good, the bad, the ugly. And I think what I'd like to do is give you an appreciation as to what AI can do for you in research, where it can perform very well, say better than advanced biostatistical tools, but where there are certainly issues and concerns in both the development and deployment of AI tools in transplant research and transplant practice. And so beyond my, say, research role, so I'm a clinician scientist, and I co-lead our transplant AI initiative, where we are both developing and deploying AI tools across the transplant program. And this is supported with critical funding from our transplant executive. So very fortunate to have that support to provide this resource across our transplant center. I'm also partnerships and engagement lead at the Tamardi Center for AI Research and Education in Medicine. And this also provides the opportunity for networking with people who have an interest in AI research as it applies to healthcare at both the University of Toronto, but also across the country and internationally. So it's a very nice way to engage and interact with computer scientists and AI and healthcare researchers. These are my disclosures, so both industry as well as current peer-reviewed grants. So first of all, I'll start off with an outline of what is artificial intelligence and machine learning and some of the, say, terminology and tools that are available and can be developed. Then we'll get into specifically transplant applications. And for the sake of this presentation, I'll give you some examples from my own research program around liver transplant. And you can take the lessons you wish to apply to your own area of research. So we'll go across the spectrum of liver transplant, so trajectories on the wait list, post-transplant patient, and graft survival. And then we'll get into some challenges that we have encountered, from a research perspective as well as from a clinical perspective. And so I can tell you that I, started working very closely with computer science professors across the University of Toronto, just as I was about to start my independent research program. And that was, say, in 2017, I started reaching out as I was still working on a PhD. On my PhD, and then we started working on various projects, some of the work of which I'll present to you today. And, from there, I would say that really my collaborations with computer science professors, as well as the co-supervision of computer science graduate students and postdoctoral fellows, has been key actually to us learning from each other. So basically, I've learned a lot from computer science trainees, as well as their supervisors through our regular discussions and interactions. And likewise, they have learned a lot from a clinical perspective. What are the nuances of the clinical questions that can be addressed with AI methods, and potentially methods that can be further created to suit specific clinical nuances. So hopefully I'll give you some insight into how we've done that across different questions of interest, and happy to further answer any questions during the discussion session. So what is artificial intelligence? So artificial intelligence is basically intelligence demonstrated by machines and computer systems that use information and can act in a goal-directed manner. And so in healthcare, certainly there are applications out there. So in terms of medical image analysis, AI-assisted robotic surgery, there are various tools that in the US healthcare system are already in use, such as medical risk prediction, fraud detection, automated workflow assistance. So there are different applications in the pharmaceutical industry. AI tools are being used to, say, optimize the performance of clinical trials as well. In terms of machine learning, so this is a subset of AI, and it's about learning equations from the data. So this allows software applications to become more accurate at generating predictive models without being explicitly programmed. And what you can see here is that deep machine learning is a subset of machine learning. And within deep machine learning is where you have generative AI, so large language models. And so deep machine learning particularly allows for leveraging hidden patterns and interrelationships within large data sets, particularly in a longitudinal manner. So longitudinal data or sequential data over time. Deep learning learns complex equations from data. So you have multiple parameters that are considered and layers that are modeled to solve complex problems. So say, for example, images to classify skin lesions, text to image generation, conversational agents, as I mentioned earlier, large language models, and protein structure. So protein structure is interesting because there was a key landmark paper published a few years ago by doctors John Jumper and Demis Hassabis, and this was the publication of a method called AlphaFold, which was to predict 3D protein structure on the basis of amino acid sequences. And this work in Nature actually had a tremendous impact on the pharmaceutical industry and various areas of research. And the impact was so significant that they were awarded the Nobel Prize in Chemistry last fall, along with David Baker. And so this is just to show you that there is a tremendous impact of AI methods developed even in the last few years that has resulted in the Nobel Prize being awarded. Certainly Jeff Hinton, who is an emeritus professor at the University of Toronto and was also at Google DeepMind for some time, was awarded the Nobel Prize in Physics in 2024 for foundational discoveries enabling the development of artificial neural networks. So really impressive areas or work that has been generated by these individuals. And certainly Jeff Hinton is certainly a Canadian success story in terms of Nobel Prize. So what are the three main paradigms of machine learning? So you have supervised machine learning, you have unsupervised, and you have reinforcement learning. So supervised learning refers to predicting labels from the data. So for example, you have a model that is trained on data that is labeled. And so this will allow you to determine for any new data point that is shown to the model, what is the label? And so, for example, you have an input of of thousands of chest X-ray images, and those are labeled as pneumonia versus no pneumonia. And then for any new chest X-ray, that supervised ML model can then predict, okay, well, this new chest X-ray has pneumonia. Unsupervised learning refers to learning representations of the data without having labels assigned to that data. And so this is to create new categories of the data. You can have, for example, say new, subtypes of liver cancer detected on the basis of transcriptomic data, for example. Reinforcement learning refers to dynamic decision making using data, and this is the type of learning that is used in gaming. So, wherein you have rewards, and these are rewards based on, say, a positive action versus, say, a negative action that is not rewarded. So, for example, an example of this would be, say, drug dosing practices. This is a figure actually taken from a review we recently published in Gut last year, and this was written by a postdoctoral fellow in machine learning on my team. And in this review, we summarize the literature around AI tools applied to omics data and liver disease. And so another interesting paradigm, especially useful with omics data, but also can be applied to other types of data, is transfer learning. So for example, if you have a small institutional data set, so of rare data, for example, in transplant, you are wanting to look at a certain condition that is relatively rare, you may want to pre-train your machine learning model on a larger data set that is out there available in the public sphere, and then bring that model to then fine-tune that model on the task with limited data at your institution. And so by applying transfer learning, this allows that model to pre-train and then further learn from the local data. Now, if we look at FDA-approved solutions used in day-to-day practice, so certainly, as I mentioned, there are different types of decision support and diagnostic support tools. There's also the implementation of early warning systems that has been reported on. So, for example, and you should take a look at this paper published a few years ago now in Nature Medicine by Suchi Sadia. And her group performed a multi-center study demonstrating that an early warning system to predict sepsis demonstrated decreased mortality and adverse outcomes. So it was a very important study performed a few years ago. So far, there are several FDA-approved AI tools, over 950 as of August 2024. 76% of them are in radiology. So AI tools are have especially been brought forth in the imaging sphere. So radiology, ophthalmology, pathology, endoscopy. These are particularly where deep ML tools can, really be leveraged to elicit important insights into a diagnosis. Another interesting, type of AI method that is emerging is multimodal AI. So this is to enable different data modalities being brought together or integrated to enable better predictive performance. So if you think about yourself in the emergency room or on the ward, you have different types of data that you're trying to bring together. Now, the reality is that the human brain can't really account for more than 4 variables at a time, notwithstanding longitudinal changes in that data over time. And so, a physician tries to integrate and bring together all these different pieces of data as well as historical data. But the reality is that it's not easy to do that and really then generate personalized predictions for the given patient in front of you. And so multimodal data fusion enables this integration of different modalities. There are different data fusion techniques, and it has been shown that it's possible to achieve better predictive performance versus a single data modality. And this particular review summarizes, this is now a few years old, but certainly there's a lot of now emerging work in this area in the healthcare space and interest in even deploying such tools into the healthcare space. So why machine learning tools in transplantation? Well, transplant patients represent a complex patient population. We have several variables that we want to consider in the care of patients who are both on the transplant list and post-transplant. And there are so many different factors, including lifestyle factors, immunosuppression, that do have a contribution towards a certain outcome. There are also complex nonlinear patterns in the different blood tests. So say, as a hepatologist, I'm constantly looking at liver enzyme patterns, liver function tests, and other blood test results to understand what it is that the given patient in front of me has as a diagnosis. Additionally, there's lots of other types of data that we accumulate over time in our electronic health records. And so my group has summarized these say, different areas, so including liver disease and transplant, solid organ transplant, AI in the setting of these in various papers in the last few years. We also published a review in the Journal of Hepatology last year summarizing the literature around AI in liver transplantation. And I think, for me as a hepatologist, what I particularly like is the idea to potentially bring objectivity to the complex decisions that we make in transplantation. And so the idea of using AI as a co-pilot for complex decision making is particularly interesting because there are various decisions that I feel could be more objectively made. And so the types of decisions or the types of tasks that AI tools could particularly be interesting in include the identification of suitable donor organs, waitlist prioritization, donor recipient matching, and prediction of outcomes. Now, of course, if the data is biased, then there's a risk of the decision being biased. But I think overall, there is a great deal of potential of bringing objectivity to complex decisions, especially if the model has been trained on a large amount of data representing the experience of multiple physicians, multiple patients. And having such a model could then be informative to bringing the most objective possible decisions to a certain task. Certainly, there have been various types of work in the past across solid organ transplantation. Dr. Alex Lupi has been a pioneer in big data and the development of AI tools in the area of kidney transplantation and transplantation. So the iBox scoring system for kidney graft survival has been an important work that is now serving as a novel clinical endpoint, clinical trial endpoint in kidney transplant. One of his key papers was in the BMJ a few years ago now, publishing on iBox, and this is really the first universal algorithm to predict the risk of kidney transplant loss. And it really considers several patient, donor, and graft parameters. This is a work in lung transplant by Shaf Keshavji and Andrew Sage and Bo Wang at the University Health Network. And in this study, they developed an AI model to predict the outcomes of ex vivo lung perfusion. So this was a gradient boosting model to integrate different features, donor, biological, physiological, and biochemical assessments to then predict the outcomes of transplant. So they considered 91 different features, and it performs very well to predict the outcomes of organ health as a result of EVLP treatment. So now I'll move on to specific research projects that my group has worked on in liver transplant across the trajectories of a patient from pre to post-transplant. And I'll start off with predicting dynamic trajectories on the liver transplant waitlist. And so within this area of work, so on the wait list, I'll start off with one particular project that right now is in submission, and it's driven by a surgical resident, Bima Husjim, who's at UC Irvine, working remotely with my team, along with Ghazal Azarfar, who's a postdoctoral fellow in machine learning on my team on our Transplant AI initiative. And the idea here was to simulate the liver transplant selection committee. So with a large language model representing each subspecialist on the selection committee. So using this concept of AI agents to then simulate the selection committee. And the idea here was that the LLM would represent a hepatologist's area of expertise, the surgeon, the anesthesiologist, the social worker. And so Ghazal basically prompted each of these specialists to provide their opinion on over 8,000 cases that were generated from the SRTR, as well as certain synthetic cases with standard absolute contraindications to transplant. And so the composite selection committee, which was a composite of the 4 large language models, then reviewed all of these recipients and was able to predict pretty accurately who were those patients who would benefit from transplant. And so this is, I think, very interesting as like a proof of concept that it is possible for an AI agent to potentially be brought in to serve as an objective opinion on the liver transplant selection committee. So then I'll move on to discussing another project that we have ongoing and is funded by the Canadian Institutes for Health Research, or CIHR, and it's inspired by inequities in liver transplant allocation. So we know that traditionally female patients have been disadvantaged on the liver transplant waitlist. Certainly with the MELD 3.0, there has been rectification in this particular disadvantage, but traditionally female patients were at 14% less chance of being able to attract an organ offer. Patients with PBC, so primary biliary cholangitis, wherein their bilirubin is the most important reflection of their disease severity. Unfortunately, the bilirubin is underweighted in the MELD scoring system, and so their disease severity is not well reflected. When the MELD score was developed initially, the patient population was hepatitis C heavy, and now we see more ALD and MASH, so alcoholic liver disease, and metabolic associated steatohepatitis. Additionally, there are various dynamics on the liver transplant waitlist, so there can be rapid changes in a patient's clinical status over a short time frame, and certainly the MELD system is unable to reflect that well. So with all of these in mind, we have been working on this particular dynamic model of end-stage liver disease for fair risk assessment. This is a project driven by a PhD student whom I co-supervised, so Michael Cooper, whom I co-supervised with Rahul Krishnan, who is a computer science professor at the University of Toronto. And what Michael did was train a deep serve model, which is a deep machine learning version of the Cox proportional hazards model, on over 53,000 patients listed for transplant in the United States since 2016. These were patients with decompensated cirrhosis without exception points, and he considered 13 static and time-varying covariates per patient. Now, additionally, what he added was a rate of change, so both the velocity and the acceleration at each time point on the wait list. And so the velocity and acceleration were also added on as features to this particular model. And when we looked at the performance of our model, which we called DynaMELD, it actually performed significantly better than both the MELD sodium and MELD 3.0 when we looked at the rank ordering of patients by their risk of death or dropout on the wait list. Then we looked at fairness, and we used a particular measure called the pooled group concordance index. And so this is the concordance over pairs where the first member is part of a defined protected group. So we were interested in seeing whether particularly underserved groups currently are helped by our Dynamelt. And so we determined that women, those with primary biliary cholangitis, those of small stature, were all would all be much better served with our system using this PGCI as a measure of fairness. Now, what is particularly interesting, I think, in order to create trust in the predictions made by a model is something called the Shapley analysis. So Shapley analysis provides interpretability of ML models. And so we can do this for the overall population, but we can also do this for individual patients when we generate those individualized predictions. So by Shapley analysis, we see that those who, the top features are creatinine, bilirubin, INR, albumin, sodium, and sex, which is reassuring because those are the top, those are the features that we consider within the current MEL 3.0. Now, what you will notice is that further down, certainly first order change or velocity of the patient's serum sodium, creatinine, albumin, bilirubin, and INR are actually pretty important for a certain subset of people. And so if we just use the MEL 3.0, then we would be underserving these particular patients. And so we can also then perform individualized interpretations dynamically and understand what are the relative weights of each of these features in the prediction. And so you can see that this can be done along the trajectory of, say, for example, a 50-year-old woman with PBC. There are different features that have different weights. So, for example, at 132 days after listing, the fact that she started on dialysis in the previous week had a more important weight than the fact that she had PBC, whereas the previous 2 times, the fact that she had PBC was a more important predictor, had a higher weight in terms of the prediction of death or dropout. And so actually we are now performing an external validation of this model on our UHN data and performing a silent trial within EPIC. So we have a data engineer through our Transplant AI initiative who is able to integrate this model into EPIC and have the data flow into that dashboard to provide those individualized predictions. And I think that's very exciting because then that'll give us a sense as to, in this silent trial, given that we have the outcomes as well of those patients on the wait list, we will then be able to determine, the feasibility of deploying such a deep machine learning tool via EPIC. And so that's still work in progress at this point. So now I'll talk about optimizing post-transplant patient survival and the work in that regard. So as you will be aware, the median survival of transplant recipients certainly needs more work. In liver transplant patients, they live 20 years less than the population life expectancy, and overall survival is compromised by a heightened incidence of cancer, cardiometabolic disease, and infection. And the care of patients is based on limited evidence. So we would like to change this and turn transplant into a cure. And so this is work that we published a few years ago in Lancet Digital Health in collaboration with Bo Wang and driven by 2 students. One was an undergraduate student in engineering and also a clinical research fellow, Amir Aji, who worked with Oswald Nitsky on this project. And the idea here was to understand and to develop a personalized risk calculator to optimize the care of liver transplant recipients. And so we considered longitudinal data over time and looked at overall survival prediction as well as survival compromised by graft failure, infection, cardiac event, and cancer. And Oswald evaluated different deep learning algorithms to predict survival. The best performing one was a transformer model, which was quite computationally heavy, but was the best performing one. And it had an overall AUC that was reasonable, so at 0.77, and then survival compromised by these different major complications between 0.80 to 0.81 for 1 year survival. And then for 5 year survival, certainly lesser performance. We then also cross-validated on our UHN data, and our algorithm had similar performance as well on our UHN data, although the top-ranked features for the overall populations were somewhat different. Now, we have been trying to bring our ML algorithms into the clinic. And so, as I mentioned, we work very closely with a data engineer, Peter Maximowski, supervised by Mike Brudno, who's chief data scientist at UHN. We've also been working with human factors design people, in addition to our standard clinical as well as machine learning team. And the idea here is to extract patient daily from Epic and embed within the hyperspace for easy access and provide ranked features. And so here, what we have is a short video just demonstrating how we are doing this within Epic. This will just give you an idea of, how it is feasible actually to integrate ML tools into Epic, and the data is basically, the back end is quite important here. So Peter Maximowski has integrated the code for the back end to allow for the free flow of data, and then you have the recommendations based on whether a patient is low risk or high risk. So it's just an example, just a test example. To get out of here. Okay. So now I'll move on to graft survival. And so, sorry, so going back to that tool, actually, we're now prospectively evaluating the performance of that tool and assessing how physicians, as well as potentially patients, would appreciate having such personalized guidance around, what are the modifiable and non-modifiable ranked features or variables that are having an impact on their health. So now moving on to graft survival, this is another paper we published in Lancet Digital Health now close to 2 years ago, driven by Divya Sharma and Amir Ajiye. So Divya developed a new machine learning method called weighted long short-term memory network. And this was developed to train a model to predict advanced fibrosis, advanced liver graft fibrosis. She trained on 29 longitudinal clinical and laboratory tests. And with this particular method, so the weighted LSTM, she was better able to handle missing data and incorporate a weight loss function to minimize bias. And so with this, it was possible to dynamically diagnose graft fibrosis, leveraging the longitudinal data with an AUC of 0.798. And these were the top ranked features for the overall population, including recipient age, primary indication for transplant, and donor age. So finally, I'll conclude with a tool that also now is under final revisions. It's called Graft IQ, developed by Divya and a clinical fellow, transplant hepatology fellow, who's now a hepatologist in Ottawa, Netta Gottlieb, worked very closely with her to develop this multi-class artificial neural network trained on longitudinal clinical and laboratory features over a week prior to biopsy. So the reality is that when we look at patients with elevated liver enzymes, we look at patterns in their laboratory data, and we try to bring those along with clinical data to understand what is the likelihood of rejection versus other causes of elevated liver enzymes. And so with this multi-class artificial neural network, there was overall an AUC of 0.866 across all the classes, with quite good sensitivity and specificity overall. And then we decided to evaluate the performance of the ML model versus the medic. So we had an experiment where we had 12 medics or hepatologists evaluate 30 cases and try to determine what was the likely diagnosis on pathology. And in fact, the ML model was better performing overall than the medic. We then looked at why the ML model was lesser performing, for example, in rejection. And I realized that it wasn't recognizing certain features that I would have, or actually all of us medics had recognized as being typical of rejection. So then we fed those clinical rules back to the model using Bayesian inference principles. So we applied that to fuse the prior knowledge provided with the clinicians along with the weighted probabilities of the ML model. And by doing this, by bringing the human into the loop to teach the model, we achieved a significantly better AUC across all the different categories, including rejection. And so finally, I'll conclude with challenges and ethical concerns. So to cover the bad and the ugly. So I think there are different challenges, including data drift, model drift over time. And so when the population changes, there are going to be changes in the performance of the model, which will degrade over time. It is also a significant issue, this generalizability of models, and that can be due to different factors, environment-specific factors, inconsistent reporting of data. And so when we see that an ML model is not generalizable in a different context, one should identify what are those factors leading to the limited generalizability and adapt that model, or perhaps even develop a new model for that different environment. Now, another challenge has been traditionally interpretability, but as I said, it is quite possible to use Shapley analysis to identify what are the key factors that are leading to a prediction. I think this is critical moving forward to instill trust in end users rather than the traditional black box nature of ML models. Hallucinations particularly are a concern. So, I think this is an area of work that still needs research, and particularly in the clinical context, because one should be very vigilant to not get incorrect predictions that then lead to incorrect management of a patient. Ethical concerns, fairness and bias. So there can be data bias and a lack of fair health data collection. There are various racial and socioeconomic factors that impact outcomes and could further be perpetuated by an ML model. And so the path forward for this is to employ methods that consider that will address data imbalance and then incorporate diverse population demographics and evaluate on fairness metrics. And so overall, I think in summary, machine learning models can be accurate at generating personalized predictions. They perform particularly well when there's several variables and trajectories of variables, hidden interrelationships. Multimodal AI will enable the integration of different data types. Certain challenges include generalizability, difficulty in interpretation, and drift over time. And so this will require prospective validation across different settings, multi-center efforts, clinical trials, to enable fine-tuning of models and ensure the best possible care of our patients. And so I'd like to acknowledge key members and colleagues with whom I collaborate on a day-to-day basis, including members of our Transplant AI initiative and key collaborators, as well as members of my research team. And welcome any trainee applications or interest in collaborating with us. And finally, I'd like to acknowledge our funding sources, particularly CIHR and the UHN Foundation. Thank you for your attention, and happy to take questions.
Thank you very much, Mamatha. That was a fantastic talk.
Thank
Um,
you.
we'll open it up to questions if anybody has any. You can either raise your hand or just unmute, put it in the chat if you'd like. I don't see anything right now, but I'll maybe start it off. And I'm curious, I mean, you touched on it a little bit about the potential generalizability of the AI tools. Um, and obviously, if there's bias in the AI because there's bias in the data that feeds the AI. And especially when we look at sort of the allocation AI tools, um, and
Hmm.
with our multi diverse communities in Canada, especially with our Indigenous populations, I think everybody would agree that there probably is the potential for some bias there. But are there any ways, and I think you've touched on some of these, but any ways on how we can minimize some of that bias to make it a little bit more generalizable, especially for the Canadian population?
Yeah, I think in the Canadian context, we lack data on race and ethnicity. It's not traditionally been collected. In the US, they routinely collect this data. So in the Scientific Registry of Transplant Recipients, it is self-reported, and that data, along with socioeconomic status, we see, at least in in that Lancet Digital Health paper, particularly socioeconomic status plays an important role, has a key predictive role in graft failure. I think it is important for us to consider collecting such data, obviously in a respectful and systematic manner, in order to inform best policies and ensure that we're doing the best we can. I think traditionally there's been concern about collecting such data, but at the same time, not collecting such data could potentially compromise the care of patients. And we don't want that either. So um, I think my sense is that racial and ethnicity data, along with socioeconomic data, is important because it's likely the intersection of those that might be that could be compromising outcomes, rather than one independent of the other. And you see that in the US context.
I
But
suspect
I
our
think
data
moving
system.
forward, I think we need to be collecting that data in order to optimize and personalize the care of patients. If we don't, then we risk not being able to provide the best care possible. Of course, there will be different opinions on this, so
Mhm.
but that's my own personal sense.
And many of the tools that you showed us are based on American data, as you alluded to. Do you
Yeah.
think we'll ever get to a situation where our Canadian data system, and hopefully we will, I hope the answer is yes, where our data systems are so strong here that we'll be able to use Canadian data to drive some of these AI tools and machine learning as opposed to always leaning on our American partners.
Yeah, so for the models that we've developed, like say the first paper that we had published, for that model, we actually tested it on UHN data. But on the UHN data, we didn't have the race and socioeconomic status in there. Um, Now, for the Canadian context, I know that there are efforts from CIHR to collect such data, starting with the transplant waitlist and hopefully with post-transplant outcomes. Specifically for our project, the Dynameld project, I think people are doing this on a project-to-project basis for the time being. So say for the Dynameld project, we have the participation of all liver transplant centers across the country for external validation of our algorithm. So we are currently awaiting data from BC Transplant, Ontario Health, and all the Transplant Quebec, etc. So across the country to be able to validate this model. So certainly on a project-to-project basis, I think this is happening. But then for this to happen at a national level, I think I'm happy that this sort of effort is now in progress with CIHR. So hopefully in the coming years we'll have access to a system that is similar to the SRTR.
Absolutely. Dr. Mustafa, you've got your hand up.
Hi, thanks so much. This was an excellent talk. My name is Ahmed Mustafa. I work with Rahul, but I work in the lab side. So I'm the clinical lab director of the HLLA. So I have a question here. So how many, I think you presented excellent amount of data, but most of the data has huge amount of data point, like you showed that you used like 50,000 patient and so on. So is there like a minimum amount of data point that you can use to generate a machine learning that can understand what we need in the future? And I will tell you the reason why I'm asking this question.
It's a good question. I think ideally you need, several variables and several patients in a study. So, at least 100 and with several data points. But I think ideally, the larger the number, the better the performance. And I mentioned this earlier, but say your institutional data set is smaller, then perhaps you pre-train the model on an external larger data set. So what many people are doing, say, is that they pre-train a model on the Cancer Genome Atlas, for example. If they do work in cancer, they pre-train their model on the Cancer Genome Atlas and then bring that model into and fine-tune it on their institutional data. Similarly, you might have a specific question that your institutional data set has some overlap with a certain population that you can derive from the UK Biobank. So perhaps you pre-train the model on that UK Biobank population and then bring that onto your institutional data set. So ideally, there should be a large number of patients because longitudinal data. Now, it could be that an AI tool is not necessary to answer the question that you're wanting to address. And it might not be the right method to use if you have a very small amount of data.
No, I think it's clear enough. We have, I think, around 650 patient data, and we are actually looking at different scoring systems. So we are looking at Matchmaker, which is an applet mismatch. The Perch, we are also doing metabolomics.
Oh, Perch.
Perch, yeah, Perch. It's a tool that predicts some sort of rejection.
Yes.
It looks at the T cell epitopes.
Mm-hmm.
Matchmaker looks at the B cell epitopes. And then we are also going to measure the metabolomics. So we have a lot of variables. And so we have all the immunosuppressive. And this data is collected for the past 10 years. So I think if you're saying 100, then I think we qualify for preparing an MLI. And I'll be very
happy
minimum.
Yeah.
To you as well.
Yeah. Very good.
Thank you.
Any other questions?